Hi dear students, hope you are all doing well and you are healthy. Um, I have sent this power to your group. You can see the powers and please tell me which power I must start for teaching. Meta Protomics. Okay, thank you. Okay, so I'll explain this picture and then okay, we must start meta transcriptomics, yes? The previous section we studied about metagenomics and we studied about um, the technique that is related with detecting DNA directly from environment and then distinguishing the bacteria. So today we are going to study the metatranscriptomics, another technique that is related with mRNA or messenger RNA. Sorry, metaproteomics, yes? I have teach metatranscriptomics too, yes? Okay, metaproteomics. Here, yes, am I right? I must teach from this power. Metaproteomics. Okay. Metaproteomics involves characterization of the protein components present in the environmental microbial community at a specific time point. It is also called community protogenomics. It determines the protein complement that is post-transcriptionally regulated and translated. The community protein complement also includes the proteins that interact within and amongst a microbial community. So you can see in this technique we must analyze the proteins and we must characterize the proteins that present in environment. So we can detect the proteins directly from environments, from microbial communities at a specific time, and then we can analyze the proteins. So this technique called protogenomics or meta-proteomics, and that is very important that it determines the protein complement. Please note, the protein complement that is post-transcriptionally regulated and translated. You know, we have some, we observe some post-transcriptionally regulated, and translated. So they are very important in bacteria, and we can realize the modifications. So it includes the proteins that interact within and amongst a microbial community. Some proteins can interact with another proteins inside the community and study them and realize them. Modifications of post-transcriptionally and post-translationally are very important in bacteria, and we can investigate with this technique. Functional microbial ecology can be studied by tracking functional genes coding for the proteins and by identifying proteins associated with stress conditions. So another important factor is stress conditions. Many kind of stress conditions are present in the natural community of bacteria, in the natural world. Okay, so we can study the functional ecology, functional microbial ecology by this method. Proteins can be separated by one-dimensional or two-dimensional gel electrophoresis to obtain the proto-fingerprint profile of the microbial community. Using this approach, proteins that are stable are readily characterized and relate directly to the genetic code. So we can use one-dimensional or two-dimensional gel electrophoresis for separating the proteins. So we analyze the proteins by gel electrophoresis and by dimensional gel electrophoresis. And then we can study the proto fingerprint of them, and then we can compare the proto fingerprint of different bacteria to each other. So the proteins that are stable are readily characterized and relate directly to the genetic code. Each genetic code can produce and can secrete a special protein. So the best method for characterizing the proteins are gel electrophoresis for investigating the proteins. Since post-translational processing is rare among prokaryotes, Proteomics provide a high throughput method for characterization of the function of microbial communities. So you can see here, I talked about post-translational processing in bacteria that is rare but it exists, and Proteomics provides the high throughput method for doing that and for characterizing the function of microbial communities. So we can study the post-translational processing in prokaryotes with metaproteomics, and then we can characterize the proteins. Okay, now in this picture you can see metaproteome characterization. You can see the flowchart at the top of the picture. In this flowchart you can see environment sampling. Like another techniques, first of all we must screen the environment. We have a screening and gathering the samples from environments, from different environments. The next step is extraction of microbial protein pool. We must extract the proteins from bacterial cells. That is not very easy. And the third step is protein separation using gel electrophoresis. I told you, for example, one-dimensional gel electrophoresis is a very good method for protein separation, as you see in the third step of this flowchart. And the fourth step is protein identification using mass spectrometry, or MS. So we can use mass spectrometry for protein identification. And then we have identification of new genes, new functional genes. That is very important for us, that each gene produces a product. We can investigate and we can realize the proteins, the products, and then the gene related to them. Identification of new functional genes is very important for us in metaproteomics technique. Okay, and then at the final we have functional analysis. So for investigating the microbial functional or functional diversity, we can use metaproteomics technique. Okay, if you have any question, you can ask. I'm waiting for your questions. If you have any question, you can ask. Okay, so I continue. Please note the previous technique was metaproteomics, and this technique is Protogenomics. They are different to each other. Please note Protogenomics mix. Okay, protogenomics based on genomic and transcriptomic information. So we have here two factors together, means genomic and proteomic. So we have two factors together to investigate this method. This method is based on genomic and transcriptomic information together. This approach facilitates in identification of novel peptides obtained from mass spectrometry-based proteomic data. So this technique is a mixed technique. We have genetic information and protein information together. So this approach facilitates the identification of novel peptides, and that is very good for us for discovering and finding a new and novel proteins or peptides or new enzymes. We can do this with a spectrometry technique. Developments in proteomics and protogenomics have been utilized to identify and provide potential treatment measures for cancer by determining proteomic markers for tumors. So here you can see developments in proteomics and protogenomics are very important to identify the treatment measures for cancers, treatment determining proteomic markers for tumors. So that is very important for recognizing and treatment of tumors or cancers. As we know, proteomic markers as proteins, so that is very important for treatment of illnesses. So please note one of the exam question, I don't know, 2 or 3 terms ago, was this, that which technique is useful to identify and provide potential treatments measure for cancers. And the students answered that the protogenomics is a very good technique for treatment of cancers, and by determining proteomic markers or tumors, we can recognize the cancers and we can treat them. So the answer of the question was protogenomic technique. Okay, it can help in better understanding of mechanisms and causative factors for various diseases and identify therapeutic targets or generate biomarkers for effective diagnosis. So protogenomics is a good technique for understanding the mechanism of illnesses, mechanism of diseases, and identify therapeutic targets. And that's maybe here as a question for the final exam. Which technique is for identifying therapeutic targets? Or which technique is useful for generating biomarkers for effective diagnosis? You must answer the protogenomics techniques. Okay, now in this flow chart, you can see Proto-Genomics Workflow. You can see the workflow here for Protogenomics. The first step is cell culture, because we want to extract the protein, we must culture the cell. The next step, we have protein extraction, and then we have peptide digestion. You know, peptide digestion is with different enzymes. And then LC mass spectrometry, or LCMS or MS, that is for realizing and identifying the activity of proteins, as I told you before. And the last step is gene annotation. So as you know, one gene is for one produce one protein, or one protein is related to one gene, so the gene annotation can be done. Okay, now you can see the tables. Okay, do you hear my voice right now? I'm going to explain the tables, so please note. Okay, I'm going to get it bigger. In this table, you can see the advantage and limitation of the Ohmic technologies. That is very important. It may be a question for your final exam. Please note and please read it carefully. So at the left of the picture and the left of the left column of this table, you can see approach or techniques, and the middle column is for advantage and the right column is for limitation. So approach metagenomics. Metagenomics, you can see here detection of complete and active enzyme clones is the advantage of metagenomics, and the limitation. You can see only information on functional approach associated with the presence of gene. With the presence of gene, another limitation is no information about their expression levels, and that is not good because we don't have any scale, any measurement for their expression level, and that is not good. Another limitation is many genes are not expressed in any particular host bacterium selected for cloning, and that is another limitation too. So we have three limitations in metagenomics technique. And because all of the products, all of the observations related to the genes, so only information on functional properties associated with the presence of gene, we can realize them. And that is another important limitation, I think. Okay, the next technique is meta-transcriptomics, and the advantage contains detailed study of the active microbial ecology possible, that is good, generates more taxonomic and potential functional data, and that is very important for taxonomy of the bacteria. The third advantage is gene expression profile at specified time periods provided, and that is very important for that we can investigate the expression of genes, not only the genes but the expression of genes related to produce messenger RNA can be realized too, and that is a good advantage. And the limitation. So we just need to isolation of RNA samples, and that is not very easy. And the keeping of RNA is not easy too. Short half-life, and that is the worst um, limitation for this technique, that short half-life of mRNA, hence difficult to detect rapid short-lived responses to environmental stimuli. So short half-life of mRNA is very, very important, and we um, are forced for producing cDNA or complementary DNA. As I told you before, uh, immediately after we isolate the RNA, we must synthesize the cDNA from that because of the short half-life of mRNA. Another limitation is presence of mRNA may not always depict presence of protein, and that is not good too, because some mRNA are antisense RNA, or they are not being expressed and producing any proteins that them. Okay, the next technique is meta-proteomics. Meta-proteomics, the advantage that contains characterizes functional protein signatures, metabolic information about microbes in the natural environmental ecosystems, and that is very good about that. It's like we are studying the microbes in the natural habitats, the natural environments, and that is a very good advantage. The limitation of this technique is contained in metaproteomics technologies. Extraction and separation for characterizing the protein complement of genome are still limited. So we have some problem with extracting of protein, extracting and separation of protein are also difficult too. So we have some limitation for doing that. Okay, the next technique is protogenomics that we read today. The advantage, it has only one advantage, and that is validation of express gene products on a large scale, which leads to correct interpretation genomic sequences. And limitation is that mass spectrometry-based proteomic studies have low sequence coverage, and that is not very good for doing that and using this uh, technique for detecting the proteins. Okay, if my sound is not good, I can exit one more time and then enter to the class. I exit for a few minutes and then enter to the dawn again. I think my voice is going to be better. I don't know, but I hope that. Okay, if you have any question until I'm entering to the class another time, you can ask me. Okay, hi again. I hope my voice and my sound is better right now. Um, one of your friends asked the question that how is the question about the uh, schedule in the exam, explanation or differential. Yes, it might be one of your exam questions. For example, enumerate the advantage and limitation of meta-transcriptomics. Okay, or maybe an important trait must be maybe answered, maybe question. For example, see this table. For example, separation or extraction of proteins are difficult in metaproteomics. Maybe ask you in which technique we have some problem with extraction and separation. For example, a distinct trait or limitation or an important advantage may be as a question. Okay. So you must read carefully. The tables are important for you. Okay, and then the modern taxonomy for microbial diversity is another title for you. We are going to start Biography of Microbial Diversity, and that is the subtitle of you as your text. Modern taxonomy for microbial diversity. The diversity of microbial communities varies within habitats as much as between habitats. So that is very interesting that the diversity within habitats is more and more larger than between habitats, and that's very interesting. This variation can even occur within a few millimeters, suggesting that microbial diversity encompasses more than the documented evidence available. So you can see many ecosystems have this variation between a few millimeters. That means in a very small niche we can see this variation, and suggesting the microbial diversity encompasses more than the documented evidence available. Hence, biogeography is gaining importance as a field of study from microbial diversity point of interest. Due to the innately small size of the microorganisms, environmental complexity plays a major role in determining diversity. I think the sound is better right now, yes? I exited from the class and then I entered, the sound gets better. Okay, so due to the innately small size of microorganisms, environmental complexity plays a major role in determining diversity. As you know, the microorganisms size are very, very small, so uh, collecting and the biofilm of microorganisms and community of them can play a major role in diversity, but not each of them alone, because they are very, very small and they are small in size. Okay, recent tools like metagenomics aid in biogeography studies by providing information on nucleic acid sequence data, thereby directly identifying microorganisms. So recent tools you can see like metagenomics aid in biogeography studies by providing information on nucleic acid sequence data. You can see many informations on nucleic acid, and many tools are designed for metagenomic uh, method, and they can be useful for gathering the information on nucleic acid and sequence data, and so we can identify the microorganisms we can. Therefore, the phylogenetic information can be used to compare microbial diversity profile across habitats. Okay. Examples of metagenomics. Sorry, I cannot understand your meaning. The example of metagenomics. What do you mean, example? Metagenomics is a technique, okay? Therefore, the phylogenetic information can be used to compare microbial diversity profile across habitats. Okay, and the last um, title in this power is microbial evolution. Microbial evolution. If you have any question, you can answer me. Okay. Uh, I explain a little about microbial evolution, and another explanation will be for the next lecture. Your teaching and your lessons are not behind, so we can just talk a little about microbial evolution, and then we will continue the next section. The evolutionary relationship of microorganisms is called phylogeny. Understanding phylogenetic profiles of microbes becomes a daunting task because of their small size and the lack of particular indicators that could serve as markers. So, in microbial evolution, we must investigate phylogenetic relationships of microbes, and because of their small size and the lack of particular indicators, that is very difficult for us to evaluate this relationship and this microbial evolution. Some proteins and genes are considered as evolutionary chronometers, which measure the evolutionary change. So we use some proteins and genes as indicators and as markers for investigating the evolutionary relationships or changes. Currently, the 16S RNA sequence is considered to be most reliable for measuring evolutionary relationships in bacteria and archaea, and the 16S sequence for fungi. However, it is necessary to choose a correct protein or gene for such studies. Such a gene or protein should have certain features which make it most appropriate for deriving evolutionary relationships. So nowadays, the 16S RNA sequence is the best and the gold standard for realizing the evolutionary relationships in bacteria, and the 16S, please note, 18S sequencing, 16S sequencing is important for fungi. So we use 16S in bacteria and 18S in fungi, and it is necessary to choose the correct protein or gene as indicator for study. We thought that some proteins and genes are appropriate for investigating and studying the evolutionary relationship or phylogenetic relationship. So we must be careful and we must be accurate for choosing the correct gene or the correct protein for investigating the evolutionary relationships. It is important to understand evolution in the context of biodiversity. Evolution leading to new ecotypes species is achieved in many ways. Some species with quick generation times also undergo mutation, frequently leading to novel species or states. As you know, dear students, the mutation is the main reason for evolutionary evolution in bacteria. You know, the mutation is needed for their evolution. So we can see horizontal gene transfer or transferring some genes by mutation between bacteria, and then they can lead to generating novel species or novel traits. Horizontal gene transfer, or HGT, horizontal gene transfer via transformation, transduction, or conjugation also accounts for the introduction of genes into distant related organisms, thereby introducing new traits and also impacting on interaction between species and thereby ecosystem processes. So, two reasons for evolution: one, mutation, and the second is horizontal gene transfer. Horizontal gene transfer is a way for changing the bacteria, for evolution of bacteria, and via transformation or transduction or conjugation, horizontal gene transfer can variate and can can participate in evolutionary relationship, can be effective in evolution relationship. It has also been hypothesized that large population sizes of microbes and their low extinction rates may also play a role in maintaining biodiversity though measurement of such extinction rates is difficult. So not just bacteria alone, but the population of bacteria, the community of bacteria can play a role in maintaining biodiversity, and that is very important for biodiversity and evolution of the world too. So your lecture is until microbial phylogeny. Please write the number, Mr. I don't know, I don't remember your name. Please write the number of power 44. We must start from microbial phylogeny for the next section, and we teach until microbial phylogeny number 44, power number 44, or slide number 44. Please write the number and the title. Microbial phylogeny must be taught for the next section. If you have any question, you can ask. I'm just sit in class for answering your questions. Okay, if you don't have any question, I will say goodbye to you, and I hope healthy and good days for you. Thank you. Bye bye.